In this lecture I will talk about how immune cells reach into the infected tissue site. So there are 4 major steps for the migration of leukocyte or WBC into the tissue. This is the endothelial cells that lining the blood vessels. So these are the blood vessels and this is endothelial cells. Beneath the endothelial cells, this is called tissue site. So this is tissue. Before the infection of tissue, there are already some immune cells are present in the tissue site. These immune cells are called dendritic cell, macrophage, mast cell and etc. Now let's assume there is an infections. If there is infection in the tissue site, let's assume these are the bacteria or protozoa or any pathogen enter into the tissue site, then what happens? This dendritic cell, macrophage and mast cell can recognize the pathogen. By it actually this all immune cells express pattern recognition receptors, so by which they can recognize the pathogen, and after recognizing this dendritic cell and macrophage starts to secrete a chemical pro-inflammatory cytokine. So they start to produce pro-inflammatory cytokine, this dendritic cell and macrophage. Example is IL, IL-1 beta, IL-6 and TNF alpha. These are the pro-inflammatory cytokines secreted by the dendritic cells in response to the infections, and this chemical actually reach to the endothelial cells. In response to this chemical like IL-1 beta, IL-6 and TNF alpha, these endothelial cells starts to produce one molecule. Before the infection there was no this molecule, but after infections this endothelial cells starts to produce a molecule called E-selectin, E for endothelial, selectin. All the endothelial cells starts to express E-selectin molecule, and when the mast cell recognize any pathogen, it starts to produce a chemical called histamine, and this molecule also reach to the all endothelial cells, and in response to this histamine, these endothelial cells also express another type of selectin molecule that called P-selectin. Through these blood vessels, blood flow, and in the blood immune cells are present like leukocytes. These are the immune cells. So immune cells go through this or flow through these blood vessels. Now this is the immune cells, immune cells. What happens? These immune cells express some ligands for the P-selectin, E-selectin, so they can bind with this molecule. When there is no infections, these endothelial cells do not express these selectin molecules, so blood flow very fast and immune cells also flow through this blood. But when there is infections, these endothelial cells express these molecules, so that now the immune cell cannot escape this molecule, so it actually halts for a very small time. And the immune cell also express a selectin that's called L-selectin, L for leukocyte, and the endothelial cells express ligand for the L-selectin. So now 3 molecules are attaching with each other, the E-selectin, P-selectin and L-selectin attaching with each other. The first step is selectin-mediated rolling. What is rolling? At first the immune cells are flow through these blood vessels very fast, but now when these endothelial cells starts to produce this type of molecule called selectin, this can bind with the immune cells, so it slows down the immune cells. But one thing I want to mention, the binding between the selectin and the ligand molecule is not very, actually the affinity is not very high, it is very low. So selectin and the ligand binding is very low affinity, because the blood flow, mere blood flow can actually detach these immune cells. So at first the immune cells bind with this molecule, but when blood flow, this immune cell actually detach from, so attachment and detachment. First it attach, then the flow of blood actually detach these immune cells into the next endothelial cells. So now the immune cells actually move from one endothelial cells to another. This is called rolling. Now these immune cells go to this next endothelial cells, it is called rolling. So this rolling is actually mediated by selectin molecule. Okay, now the immune cells present is here now come into this cells, and again it bind with the molecule. The immune cell also express a L-selectin that bind with these endothelial cells. But another thing occurs when these immune cells recognize the pathogen. It also secrete one type of chemical that's called chemokine. This is very important, and this chemokine go to the immune cells. These immune cells express some receptor for chemokine. So these are the receptor for the chemokine, and chemokine come from these immune cells and bind this receptor. Chemokine bind with the chemokine receptor. What happens? These immune cells actually express integrin molecule, but when there is no chemokine, the integrin molecule is bent in conformations. These are the integrin molecules already present in the immune cells. This can be LFA1, this can be VLA4. These are the integrin molecules. So another important molecule is integrin. Example is this and this. So when there is no chemokine, the immune cell express this integrin but in bent conformations. But when this chemokine bind with the receptor, the integrin become extended in conformations. Now it becomes like this. It becomes like this. Extended conformation, and in this extended conformations, the endothelial cells express actually receptor that can bind with this molecule integrin. Receptor that bind with the LFA1 called ICAM, and the receptor that bind with the VLA4 called VCAM. Intracellular adhesion molecule and vascular cell adhesion molecule. So the second step for the migration is chemokine-mediated increase in affinity of integrin. So this is the first conditions, rolling. The second condition is increasing affinity of the integrin. Now the third situation occurs. The third point is, as the integrin become high in affinity, what happens? Integrin-mediated arrest. Integrin-mediated arrest of immune cells occur. So now the immune cells cannot flow. At first it was rolling because the selectin and the ligand for the selectin, the affinity between this binding is low, so it can roll down. But when chemokine binds with the receptor, the integrin affinity increases. It binds with permanently with the endothelial cells. It becomes arrested. So it is called arrest. So immune cells become arrested. So this is the third step in which the immune cells now attach with all the molecule present in the endothelial cells. Now you can see the immune cells is binding with the selectin ligand and the integrin receptor. So now it becomes arrest. And the fourth step then occur is called transmigration, or you can say it diapedesis, or you can say extravasation. But there is a problem. Why? Because these endothelial cells are tightly joined with each other, with every cells. They tightly joined with each other. These are called adherence junctions. So how could these immune cells go through these adherence junctions? So immune cells express one molecule called CD31 that actually disrupt this molecule, disrupt this adherence junction transiently, not permanently, and then the immune cells can enter into the tissue site. Now these immune cells actually entering into from the blood vessels into the tissue site. This is called transmigration or diapedesis or extravasation. So these are the 4 steps by which immune cells reach into the infected tissue site.